Good afternoon to all the BIPC second year students. I hope you are in midst of your IPE, MCET and NEET preparation. There is nothing to worry with, wherein you have to grow with your concept and be ready both for subjective and objective type of answering questions. Okay. So, as such, as we have dealt with all the long answer type of questions, short answer type of questions, now we have landed up with the chapters that is from plant physiology. Today we are going to cover up two chapters, that is transport in plants and mineral nutrition from plant physiology. Most repetitive questions. From these questions, how you can cover up your MCET and NEET also I will be telling you. So, here if you see, how does ascent of sap occur in tall plants? How does it occur in tall plants? This question has appeared in multiple years, like you see in May 2024, 2018, 2015 and March 2015 as well. In these many years the questions have appeared. So, here if you look into the answer, ascent of sap, of course transport in plants is deleted in your updated NEET syllabus, but then transport is in plants is important concept for your MCET. Okay. So, you have to remember here how the transport is being done and what is the role of symplastic and apoplastic pathway, how is it driven in the endodermis, everything you have to learn in detail and then have a schematic presentation of your answer. Okay. So, if you see here ascent of sap, about ascent of sap, first you give out a proper definition about ascent of sap. Ascent of sap is nothing but what? Upward movement of water and dissolved mineral salts from where? Root hair, from root hairs, then the water and minerals move to root cortex, followed by they will move to xylem, stem and leaves. Okay. From root hairs to root cortex, then to xylem, then to stem and then to leaves. Through tracheary elements, this is through what? Tracheary elements, tracheates and vessels of the xylem. Here if you see, root hairs will absorb water from soil, this is by the process of osmosis. Okay. Osmosis, which is from high concentration to low concentration, as it is to along with the concentration gradient, it is not against the concentration gradient, it is along the concentration gradient. Fine, right. So, this is across the concentration gradient. So, root hairs will absorb water and minerals from the soil through osmosis process. Water enters the xylem of roots, first it will enter into what? Root hairs and then it moves upwards across the xylem vessels, from the root tip to the tip of the plant it will be moving up. Finally, it reaches the leaves and growing tips, where it is utilized for the process of photosynthesis and other metabolic activities. So, this is the brief intro that you will give about ascent of sap. Now, you have to clearly mention what is the main driving force for ascent of sap. Main driving force for ascent of sap will be transpiration pull. Okay. Transpiration pull is the main driving force for the movement of water and minerals from soil or from root hairs to the tip of the plant, where transpiration is nothing but loss of water vapor. It is loss of water vapor from leaf stomata, which creates tension or negative pressure, that is called as suction force in the leaf xylem. Okay. This is because of what? This is because of transpiration pull. As water is being transpired from the leaf surface, what happens? Negative pressure or suction force is built, wherein the water is pulled from the leaf xylem. This negative pressure will pull the continuous water column upwards from roots to leaves. Clear, right? Next is physical properties of waters. What are the physical properties of water that is supporting the ascent of sap? Main physical property is cohesion and adhesion. If you see cohesion, what is cohesion? Cohesion is nothing but mutual interaction between water molecules, which you can, where you can also address it as mutual attraction between water molecules. Okay, this is mutual attraction between water molecules, where it keeps the water column continuous. Next is adhesion. This is attraction between water molecules and walls of xylem vessels. Okay, this is attraction between water molecules and walls of the xylem vessels, which will help the water to rise in the narrow tubes. Then tensile strength and capillarity. What is tensile strength and capillarity? This will enable water to resist breaking. This will enable water to resist breaking under tension, and this will rise in the thin xylem tubes. Next, in the young herbaceous plants, ascent occurs via what? Primary xylem, not secondary xylem. This is in young herbaceous plants. In woody trees, you have tracheids and vessels which are well developed, that is occurs in what? Secondary xylem. Remember, in herbaceous plants, young herbaceous plants, ascent is through primary xylem. In woody trees, the ascent or movement of water is through what? Secondary xylem. So, initially you will explain about ascent of sap, that is transport of water from root hairs, root cortex, xylem vessels until tip of the plant, and then you will clearly mention what is the driving force of the ascent of sap. The driving force of ascent of sap will be transpiration pull, and then you will mention about the physical properties of water, highlight cohesion and adhesion of water molecules, and then mention about tensile strength and capillarity of tracheids and vessels, and how the ascent of sap is driven by xylem vessels, primary xylem in herbaceous plants and secondary xylem in woody trees. This is how you have to present your answer. Okay? Now, properties of water are important even for your MCQs. You may get statement based questions as well. As such, you prepare the concept in detail. For subjective answer, you have to have a mind map on how to present the answer. For objective, be ready with all the statements and the concept. Okay? This is how you have to go through each answer. What is meant by plasmolysis? How is it practically useful for us? Asked quite a times like in 2018 and 2016. First, you have to mention out what is plasmolysis. Okay? What is plasmolysis, that you have to mention in detail. So, here if you see, plasmolysis is nothing but the shrinkage of cytoplasm and plasma membrane away from the cell wall. A plant cell, when a plant cell is placed in hypertonic solution. What is hypertonic solution? For example, if I have to take a resin, okay, this is a beaker, let us suppose this has a beaker wherein complete molecules, water is present. This water is with sugar solution, sugar or salt solution, fine, right. This complete water is having what? Sugar or salt solution, which is highly concentrated. If you keep a resin into it, okay, when you keep a resin into this mixture, fine, right. This is of course a plant cells are present, wherein cell wall will be there, right. Yeah. So, cell membrane and cytoplasm will shrink away from the cell wall. This will shrink away from the cell wall. As such, the resin which is in this condition will be shrinked to this condition. Fine, right. So, what is plasmolysis? The shrinkage of cytoplasm and cell membrane away from the cell wall, that is towards the cytoplasm, it will shrink when it is placed in the hypertonic solution, that is concentrated sugar or salt solution. Water moves out of the cell. This is because of what process? Exosmosis. Exosmosis, where the water is moving from low concentration to high concentration, which is across the concentration gradient. As concentration is high in the sugar solution, low in the resin, the molecules are moving, water molecules are moving from the resin towards the solution. Okay. So, water moves out of the cell, which is addressed as what? Exosmosis, through the plasma membrane, which is causing what? Protoplast to contract. What is protoplast? Protoplasm along with cell membrane, you will address it as protoplast. Remember, what is protoplasma? Protoplasm along with cell membrane. Clear, right? Next, you see stages of plasmolysis. First, normal cell. Vacuoles are filled with cell sap and protoplast is pressed against the cell wall. That is how protoplast is contracted. First sign of plasmolysis is addressed as incipient plasmolysis. Incipient plasmolysis means first sign of shrinkage. This is the first sign of shrinkage wherein protoplast just begins to pull away from the cell walls at the corners. Okay, protoplast will just pull away from the cell wall at the corners. This first incipient plasmolysis and then which is followed by complete plasmolysis. Okay, complete plasmolysis where protoplast is fully contracted and it detaches from the cell wall, leaving the gap filled by external hypertonic solution. What are the practical uses of plasmolysis? Now, you have clearly understood what is plasmolysis. Plasmolysis is because of what? Exosmosis, that is happening when cells are placed in hypertonic solution. Best example I can give you is dropping out a resin in highly concentrated sugar or salt solution, wherein protoplast, which includes cytoplasm and cell membrane, that will contract out of the cell wall, where the first sign is what? Incipient plasmolysis. Next will be complete plasmolysis, where a gap is filled in between cell wall and cell membrane. That gap is filled by what? External hypertonic solution. Clear, right? Next is what? Practical uses of plasmolysis. What are the practical uses of plasmolysis? First, in the food preservation. Pickles, meat, fish are preserved by salting. Okay, pickles, meat and fish, they are preserved by salting. Jams and jellies are preserved by high sugar concentration. Both will create hypertonic conditions that will kill bacteria and fungi. This is induced by what? Plasmolysis. Next use is weed control. Next is for experimental studies in laboratories to demonstrate osmosis and cell membrane permeability. This will be used. Exosmosis process or plasmolysis principle is used. Fine, right? You have to mention what is plasmolysis by giving out a clear example. Steps of plasmolysis you have to include about incipient plasmolysis and complete plasmolysis, and then highlight about practical uses of plasmolysis. Clear about food preservation, weed control and experimental study. Clear till here, ma? So, this is one diagram which is for the previous question where we have learnt transpiration pull, right. Next, here is a question that transpiration and photosynthesis is a compromise. Explain about it. Transpiration and photosynthesis is a compromise. Explain about it. One time asked question. For this you have to clearly explain on photosynthesis requires water, whereas transpiration leads to water loss through stomata. Photosynthesis, one of the raw material for photosynthesis is water, but transpiration will lead to what? Water loss. That you have to mention. Transpiration pull will provide what? Driving force for upward movement of water. Water supply, it will maintain continuous water flow to mesophyll cells for photosynthesis. And next, transpiration will provide what? Cooling effect on leaves. Fine. It will reduce the leaf temperature, whereby protecting the enzymes which are present in the mesophyll cells. Next is about turgidity. This will maintain cell turgor, keeping the leaves expanded for efficient photosynthesis. This will keep the leaves expanded for efficient photosynthesis. Next, high water need. High water need where active photosynthesis will increase the dependence on transpiration. This will increase the dependence on transpiration. Next is limiting factor. Excessive transpiration will cause what? Stomatal closure, thereby reducing carbon dioxide entry and carbon dioxide fixation. What is the ecological impact because of transpiration? Large scale of transpiration will maintain what? Atmospheric humidity. Atmospheric humidity is maintained by large scale of transpiration. Next, C4 adaptation. Okay, C4 adaptation where C4 plants are involved to fix more carbon dioxide with less water loss. This is how transpiration will aid photosynthesis but competes for water. So, two processes exist in compromise. For this, what are the reasons that you are highlighting? That is important. First, you are explaining about transpiration full, then water supply where you are highlighting ascent of sap, and then cooling effect because of transpiration, turgidity, high water need, limiting factor because of excessive transpiration, stomatal closure will happen, thereby it limits the intake of carbon dioxide, thereby it will limit the carbon dioxide fixation. And then ecological impact. What will be the ecological impact if there is large scale of transpiration occurring? It will increase high humidity levels. And then if you see C4 adaptation, what is C4 adaptation? In respect or with respect to transpiration. Then these are the main points to be highlighted, where at the end you will say both the processes compete for water, so this exists as a compromise. This is how you need to highlight and explain the answer. Next, explain the mechanism of opening and closing the stomata. I first recommend you to draw the diagram and then you start explaining about opening and closing a stomata. First you see stomata are small pores. First explain about stomata and stomatal apparatus. Okay. Stomata are small pores on the leaf surfaces. These are surrounded by two guard cells. These are surrounded by what? Two guard cells. Fine, right. The opening and closing of stomata will depend on the turgor pressure of the guard cells, which will act as valves regulating gaseous exchange and transpiration. Daytime usually stomatal opening will take place, which is addressed as photoactive stomata. Daytime stomatal opening, which is addressed as what, ma? Photoactive stomata. Fine, right. So here the ion movement first thing, light will trigger active transport. Ion movement, if you see, light will trigger active transport of potassium ions into guard cells from surrounding subsidiary cells. Next, water potential will decrease as such. Influx of potassium is accompanied by passive uptake of chlorine as well, chlorine ions as well, thereby lowering water potential of guard cells. Osmotic water uptake. Water enters the guard cells by osmosis. This will make the guard cells turgid. Aperture formation. The thin outer layers will bulge outward, pulling the thick inner walls apart, opening the stomatal pore. So, this will be the inner walls. Yeah. So, here if you can see stomatal opening, what is happening? Intake of potassium ions, by passive movement of chlorine ions as well, and then water movement inside into the guard cells from subsidiary cells is making the guard cells turgid, wherein outer walls will be thin and inner walls will be thick. Fine, right? This will help in the opening of stomatal pore or aperture. Fine, right? Which opens the stomatal pore. This is how stomatal opening is taking place. If it is opening during daytime, it is addressed as photoactive stomata. If stomatal opening is during night time, that you will see in CAM plans, that is addressed as scotoactive stomata. Here we go with nighttime stomatal closure. Fine, right? How stomatal closure will happen? Ion efflux. Here you have observed during the stomatal opening, ion influx will take place. That is, both potassium and chlorine are entering into the guard cells. This is influx of ions. If you see stomatal closure, in darkness what happens? Efflux of ions, potassium and chlorine ions, that is efflux of ions which will exit the guard cells. This will increase the water potential, where water loss also will happen from the guard cells. Fine. fine, right? This will move out of the guard cells by osmosis, reducing their turgor pressure. So, loss of turgidity is happening over here. Because of loss of turgidity, guard cells will become flaccid. Guard cells becomes flaccid and this will be unable to keep the pore open. Fine, right? Lastly, closure of the pore. How the stomatal pore will be closed. The elastic inner walls will return to their original shape. The inner walls, elastic inner walls will get back to their original shape. This will cause stomatal aperture to close. Fine, right? This will cause the stomatal aperture to close. So, the opening and closure of stomata are entirely controlled by turgor changes in the guard cells. Therefore, stomata act as turgor operated valves. Remember this term, very important. This is turgor operated valves. Fine, right? That regulate transpiration and gaseous exchange in the plant. So, you have to clearly explain the mechanism of stomatal opening and closing. It will be very better, far better if you are highlighting the terms like photoactive stomata and scotoactive stomata based on the time they are opening. This is the complete details about what? Stomatal opening and closure. Next we move on to transpiration is a necessary evil. You have to explain this statement. Multiple times asked question like in June 2023, 2020 and March 2016 or 2017. Here if you see, transpiration is nothing but what? Loss of water as vapor from aerial parts of the plant. This is from aerial parts of the plant, mainly through stomata. It plays dual role. It plays dual role through indispensable for plant life. It can also be harmful under certain conditions. Because of these contrasting effects, it is the scientist Curtis who has described transpiration as what? Necessary evil. Fine, right? Now you have to mention the advantages of transpiration and disadvantages of transpiration in detail. Okay, just remember the subheadings or side headings in detail and give out a proper explanation using proper terminology. Need not to muck up the answers beta. Just remember what are the points to be highlighted, that's it. Okay, and mention it in sequence in a systematic way. That's it. There is no requirement to mug up the answer. Here you see what are the advantages of transpiration. Transpiration pull for water transport. This is one advantage because we have learned how come transpiration pull is helpful. This is the driving force for ascent of sap. Cohesion, adhesion, water interactions will be increased. This will give cooling effect to the leaf. These are the points you can include over here. Okay, transpiration pull for water transport. Second, mineral transport and it provides what? Cooling effect. Fine, right? Next, maintenance of turgidity and structure. How come it is maintaining the turgidity and structure? These are the 3 points to be highlighted under advantages of transpiration. Now we will look on to the disadvantages of transpiration. Disadvantages of transpiration where increased water demand. How water demand will be increased? Plants engaged with active photosynthesis, they will lose large amount of water. They will lose large amount of water. This requires continuous absorption of water to compensate the water loss. Second, limitation of photosynthesis. How come photosynthesis is limited? Next, risk of leaf wilting and leaf shedding under stress because of over transpiration pull or because of transpiration, continuous transpiration. What happens? Leaf will shed and leaf will wilt under stressful conditions, especially heat stress. So, these will be the major disadvantages of transpiration. Remember the points and highlight. It is okay if you are presenting it in the form of a table also, like on the one column if you are listing out the advantages of transpiration, on the other column if you are listing about the disadvantages of transpiration, that is also fine. Now, we will move on to define and explain water potential. How water potential what is water potential? You have to define and explain it properly. Water potential is nothing but movement of water, measure of free energy of water molecules. Potential energy, you can say, measure of free energy of water molecules in a system compared to that of pure water under standard conditions, under standard temperature and pressure. That is addressed as what? Water potential. During water movement, it determines the direction in which water moves always from a region of high water potential to lower water potential. You can also say high concentration to low concentration. Sorry, low concentration to high concentration. Okay, low concentration, wherein high water potential means low solute concentration will be there, right? That is low concentration to high concentration. Fine, right? Low to high I am talking with respect to solute, and water potential I am talking with respect to water, high water potential to low water potential. Water potential is expressed in the form of what? Pascals. What is the measured unit? Unit of measurement over here for water potential is pascals, and this is denoted by a Greek symbol of psi. Fine, right? The water potential of pure water at standard temperature and standard atmospheric pressure is defined as 0. Okay. Water potential of pure water will be 0. Effect of solute. Adding solutes which will lower the free energy of water molecules, because some water will bind to solute particles, reducing their availability for water movement. Fine, right? And now solute potential. This component of water potential becomes more negative by increasing what? Solute concentration. If you increase the solute concentration, water potential will get negative. Pure water solute concentration will be 0, and in solutions it will always be negative, remember. Now about pressure potential. Pressure potential in a turgid cell which is completely occupied with water. In turgid cell it will be more than 0. In a flaccid cell or a plasmolized cell, just now we have learned plasmolized cell, the pressure potential will always be equal to 0. So here if you see total water potential, how you can calculate the total water potential? You can calculate the total water potential by water potential is equal to solute potential plus pressure potential. Fine, right? Role in plants. What is the role of water potential in plants? Water potential will control processes such as osmosis, nutrient absorption, transport of water, and it will maintain turgidity in the cells. Now here we will see the key concept. Water moves spontaneously from region of high concentration to lower concentration. This will enable essential physiological activities in the plants. Clear till here? This is the complete and brief explanation about water potential, where you will highlight the movement of water from higher water potential to lower water potential. What will be the water potential? How is it measured? By what units? Okay, what is it for pure water? What is it for water solutions? And how solute potential is there? What is pressure potential? How you can calculate total water potential? What is its role? This and all are important. This is how you have to present your answer. This is the complete set of questionnaire from transport in plants. Now we will move on to mineral nutrition. Mineral nutrition also, the first question have put from transport in plants only, that explain how plants absorb essential elements. How plants will absorb by apoplastic and symplastic movement. You have to highlight what is apoplastic movement and what is symplastic movement. This diagram is mandatory. If you are presenting that answer, this diagram is mandatory. Apoplastic movement is transport of water. See, first from the root hair epidermis, water is transported both in the intracellular and in between the cell walls. Movement of water through the cell walls is addressed as apoplastic movement. This is addressed as apoplastic movement. Movement of water across the cytoplasm of the cell, this is addressed as symplastic movement. So we have learnt in the anatomy that the roots of the endodermis, in 11th standard we have learnt, right, wherein endodermis cells will have suberin depositions in its radial and tangential walls. Endodermis has what, ma? Suberin depositions in radial and tangential walls. Fine, right? And this is how from endodermis apoplastic movement is stopped and symplastic movement is continued. Okay? And this is how water enters what? Pericycle, from pericycle to xylem tissue. Fine, right? This is the complete explanation of what? Apoplastic and symplastic movement. Explain it in sequence like this. If you see, okay, just go through this diagram. If you are having any doubt, just ask me. Here we go. Yeah, apoplastic movement. In this pathway, ions will move through the cell walls, ions move through cell walls and intercellular spaces without crossing any cell membrane. This is without crossing any cell membrane. It involves what? It involves free space or outer region of the cells, that is outside the plasma membrane. Movement of ions is passive. Always remember, movement of ions is passive, that is it occurs along or across the concentration gradient. It occurs along or across the concentration gradient without using any energy. This process allows rapid uptake of ions. This allows rapid uptake of ions, and usually this occurs through ion channels which are present in the cell wall. Next is symplastic pathway. In this pathway, ions will move across the cytoplasm in between the adjacent cells. Movement is slower as compared to the apoplastic movement. Remember, the transport of ions in the symplast always occurs against the concentration gradient. This is always against the concentration gradients. Therefore, it is an active process that this will enable selective absorption of mineral ions. Fine, right? This will enable selective absorption of mineral ions. So, here if you see apoplastic pathway, symplastic pathway, this you can explain in an organized way entitling to two titles, or you can also give a tabulated presentation where you are explaining about apoplastic and symplastic pathway by comparative analysis. That hits high. Okay? So, in that way also you can explain, but remember that this diagram is mandatory. Mandatorily you have to present this diagram. And next, write in brief how plants synthesize amino acids. Quite a times asked question again. How plants will synthesize amino acids. Just a minute. Yeah, here is the answer. You see it carefully. Yeah. Plants will synthesize amino acids mainly by assimilating nitrogen. This is mainly by what? Assimilating nitrogen. Fine, right? In the form of ammonium ions. These are derived from nitrate reduction or nitrogen fixation. Fine, right? Ammonium ions are then incorporated into organic compounds to form amino acids through two main processes. First one is reductive amination, second one will be transamination. What is reductive and what is transamination, you see here. In reductive amination, what happens is ammonium ions will combine with alpha ketoglutaric acid in the presence of the energy reducing energy, that is NADPH, and the enzyme glutamate dehydrogenase. This is the enzyme. This gets converted to what? Glutamate, water, and this will be oxidized to NADP plus. Here NADPH is acting as what? Reducing agent. Remember the reaction first. Okay. If you remember the reaction, then you can easily explain. Fine, right? Alpha ketoglutaric acid with ammonia and NADPH in the presence of the enzyme glutamate dehydrogenase. This will give out the products like glutamate, water and NADP plus. This is the primary step where inorganic nitrogen is converted to the organic form. Next, transamination. In this process, amino group from one amino acid is transferred to keto group of the new amino acid. The reaction, if you see, if this is amino acid 1, keto acid 2, fine, right? Amino group will be transferred by the enzyme transaminase and this forms keto acid 1 and amino acid 2. Best example I can give you is glutamic acid, which acts as the main amino donor. This is by what? Transferring the amino group to form various other amino acids. This is the clear note of this, fine, right? Now we will move on to the nitrogen fixation in the leguminous plants. How the nitrogen fixation occurs in the leguminous plants. This is again an important question. Diagram is mandatory. Firstly, what are root nodules? How root nodules are formed? Root nodules are the special structures which are formed from the roots of leguminous plants. This is by symbiotic association between gram-negative bacteria, that is Rhizobium leguminosarum bacteria in legume plants. These nodules will help in biological nitrogen fixation. The processes involves each and every step is important. See, I am telling you for MSET students, whoever are writing MSET exam, nitrogen metabolism is very important, ma. You have to remember all the steps in detail. They can ask you to arrange the steps in sequence. They can give out or list out 5 statements and ask you to arrange it in sequence. Not just for your IPE exam, that is important for your MSET also. So, remember it in the sequence. First is attraction of Rhizobium bacteria. How the Rhizobium bacteria is attracted? That is by release of some amino acids and sugars from root hairs to the bacteria. And then the bacteria gets attached to the epidermal cells of the root hair. Infection of the root hair. This forms a hook. As it is infected by bacteria, this forms a hook. And bacteria travels into the cortex region. This is how the formation of infection thread happens. Then entry of bacteria into the cortex region. Lastly, formation of bacteroids like this. This formation of bacteroids only, we are addressing it as what? Root nodules, where nodule formation is taking place at the end. The division and growth of the cortical and pericycle cells will help in the formation of root nodule. This involves cell differentiation and development of distinct structure. Clear till here? Any doubt anywhere? Fine, right, ma? So, this is the complete information about short answer type of questions in connection with your MSET and NEET. Of course, these 2 chapters are not there for your NEET, for your MSET. Okay? Remember, learn the concept in such a way, again and again I am telling you, where you will be ready for subjective and objective exam. Okay? Do not just mug up the questions for IPE exam. That will not be a good or wholesome preparation. Okay? So, that is all for today's session, ma. In the next session, we will solve out or will figure out few more short answer type of questions from plant physiology. Okay? So, that is all for today's session and thank you.